Today we will be explaining the protein insulin. Insulin is found in the pancreas of the human body. It is used to help regulate blood sugar. Our blood sugar changes frequently based on food intake and exercise, but remains in a range to stay healthy. Insulin works with glucagon to regulate blood sugar so it can stay in the range. Insulin gets released in the beta cells of your pancreas after eating to help break down the sugars. There are roughly 200 units of insulin in the average adult, and 30 to 50 units get released daily. It gets released into the bloodstream and then makes its way through the body. The biggest thing about insulin is that people who lack the ability to make it themselves suffer from diabetes. Let me give you a brief history of it. Starting off in 1889, two German researchers found that when the pancreas gland was removed from dogs, the animals developed symptoms of diabetes and died soon afterward. This led to the idea that the pancreas was a site where they called pancreatic substances, but what we now know as insulin was produced. Moving on to 1910, Sir Edward Albert Sharpe Schaefer suggested only one chemical was missing from the pancreas in people with diabetes. This chemical was insulin. This discovery really helped people with diabetes. Before insulin was discovered, diabetes patients didn't live long because there was no way to control how the sugars in your body were broken down. In 1921, a young surgeon named Frederick Banting and his assistant Charles Best figured out how to remove insulin from a dog's pancreas. This helped a dying 14-year-old with diabetes in Toronto. In January 1922, Leonard Thompson became the first person to receive an injection of insulin. Within 24 hours, his dangerously high blood glucose levels dropped to nearly normal levels. In 1923, Banting and McElroy received the Nobel Prize in Medicine, which they shared with Best and Collip. Later that year, Eli Lilly started a large-scale corporation of insulin that was able to provide for the entirety of North America. This led to Novo Nordisk Pharmaceutical Inc. manufacturers developing slower-acting insulin in 1936. But insulin from cattle and pigs were used for humans, and many had allergic reactions. Finally, in 1978, the first genetically engineered synthetic human insulin was created using E. coli bacteria. Shortly after that, Eli Lilly went on to sell the first commercially made biosynthetic human insulin under the name of Humulin.
Now that you know a little bit about insulin, let's go further into the protein itself. The insulin globular structure can look like this. It seems confusing looking at it, but through a ball stick model it seems to be less confusing. But to make the diagram even easier to read, we can look at this basic diagram. It's not that confusing after all. There are only 51 amino acids in the entire structure of insulin. This makes it a pretty small protein compared to many others. And there are reasons for this. Insulin is made up of two protein chains that are connected through disulfide bonds. The third disulfide bond causes a loop within a single chain. These bonds are connected with cysteine. Cysteine seems to be the most important amino acid within the protein because it is the only amino acid that has a job, and that job is to connect the alpha helixes with disulfide bridges. You can see this more in the 3D diagram. These coiled strands are the alpha helixes, and the yellow strands represent the disulfide bridges. You can see them connecting two of the strands and folding over itself on the other. Disulfide bond formation involves a reaction between the sulfhydryl side chains of two cysteine residues. A sulfur anion from one sulfhydryl group acts as a nucleophile, attacking the side chain of a second cysteine to create a disulfide bond, and in the process releases electrons, reducing equivalence for transfer. If we look back at the structure, we can assume that these amino acids are cysteine because they are the ones creating the disulfide bonds. It is also polar, which makes sense why it can be found on the outside. Another major amino acid is leucine. It is a dietary amino acid with the capability to direct stimulant myofibrillar muscle protein synthesis as well as induce insulin secretion. It plays an important role in the regulation of mTOR activity, which provides energy to investigate if a leucine sensing mechanism in the mTOR pathway is involved in insulin secretion. It is a non-polar amino acid, so it would most likely be located on the inside of the cell. Another amino acid found in insulin is alanine. Metabolism of alanine has been found to enhance insulin secretion from isolated islet and clonal beta cells of human pancreas. It is a non-polar and hydrophobic, so it would definitely be found on the inside of the protein. Arginine is also found in insulin. It improves the burning of glucose by as much as 40%. It also stimulates production of glucagon-like peptide, or GLP-1. an intestinal hormone that regulates appetite. This does not mean people with type 2 diabetes should go out and buy arginine supplements. Dietary arginine is better. This can be consumed through nuts, seeds, legumes, mostly protein products. Arginine is also polar, meaning it can be found on the outside. Here is a diagram of the polar amino acids versus the nonpolar ones. Like it says, nonpolar are normally found on the interior and polar are found on the exterior. Now, all of these amino acids have a function to help the protein of the insulin do what it is supposed to do. But like I mentioned before, the cysteine that creates the disulfide bridges is important because it makes the structure of insulin. Since there are two disulfide bridges connecting chain A and chain B, it causes them to come together and get smaller. Same with the third bridge folding on itself, making it rounder and smaller again. Insulin is categorized as a hormone which has to travel through the bloodstream. Smaller hormones can travel faster, which is why insulin needs to be small, which is very helpful in regulating blood sugar. This is how the shape directly correlates with its function.
Like said before, insulin is mostly used for diabetes. Human insulin has become a medicine for diabetes. It can be used for both types. Type 1 diabetes is when the pancreas cannot produce any insulin, and type 2, where the blood sugars are too high for the amount a body secretes is not enough to level it out. There are different types of insulins that can be picked from to work best in the particular body. Humalog, Humulin RU100, Novolin R, and Novolog are some examples of these. There are many more varieties based on need. They can be injected under the skin multiple times a day in order to maintain the blood sugar level compared to a person who creates their own insulin.
Scientists are thinking of other implications insulin can be used. Insulin can be directly applied to the skin to enhance the healing process of wounds. Adding insulin at the ratio of 1 unit of insulin per 11 grams of dextrose in patients with diabetes receiving TPN containing 150 to 300 grams of carb per day is an effective initial step to prevent and reduce hyperglycemia. A test done on fruit flies concludes when the chemical messages sent by an insulin-like hormone are reduced inside the fat cells of the fruit fly, the fly's lifespan increases significantly. Bodybuilders take insulin to increase stamina and improve endurance. It's also used to increase muscle bulk as it stimulates glycogen formation, which feeds the muscles during a workout. Insulin works to prevent the breakdown of muscle protein, which also helps a person bulk up. Insulin therapy could be harnessed to better satiatemia, reperfusion injury, and weaken the inflammatory processes heightened by the hyperglycemia and associated with brain death, and subsequent organ rejection and infection. Septic shock can be managed by control of hyperglycemia and sepsis and is considered to be a very effective therapeutic approach. As well as other critical illnesses in the intensive care unit, the body cannot produce enough insulin to keep up what is going through. These are just a few examples of the extensive research scientists have done with insulin.
Insulin is constantly being researched, and new explorations are being discovered. Scientists hope to cure diabetes one day. But for now, thank you for listening to the basics of insulin.